let's start our today's session. In the last session, we had discussed about the basic terminologies related to the genetics, right? And Mendel's contribution. So who will tell me the Mendel's contribution? Kevin will tell me. Yes, Kevin. Yes, Kevin. Am I audible? Sarjana.
Ma'am, Mendel is basically recognized as the world's as the father of genetics, and he has taken a step to study about the inheritance, how the the trait is passed from the parents to the offspring. So he conducted an experiment using pea plant. He noted all the observations by conducting the experiments, and he gave monohybrid cross, and he said about F1 generation, F2 generation, and it was he gave the Punnett square, and he gave many observations about.
Oh, Mendel gave Punnett square.
No, ma'am. We studied about Punnett square, doing the while studying the inheritance, ma'am.
Okay. Okay, everyone, please look at here. So they are the father and the mother, and they produces the baby, right? So the parent actually inheriting their characteristic, right? Though the offspring is getting all the characters from their parents, but still they are not the exact copy of their parent, right? They have some uniqueness. We all have some characters from our father, from our mother, but still we are not exact copy of either of the parent, right? Why? Because in sexual reproduction, there is a fusion of male and female gamete, right? And because of this fusion, recombination takes place, recombination of genes, right? And because of this recombination, what happens? Variation takes place. So here the mom's gene is like capital C and capital C, and the dad's gene is, it should be small c, okay? So during the crossing or the fusion, the combination will be like in the first generation, okay? The heterozygous one, right? So in the heterozygous condition, the dominant one, the gene or the allele who is actually expressing themselves completely and suppressing the other's activity, right? So only the expression will take place in the next generation, only the dominant one. For example, the hair color, okay? So this is what inheritance actually variation of traits. Why? Because of the sexual reproduction or the recombination. So in today's class, we are going to understand about the inheritance of gene. Okay? So first we need to know what is gene. What is gene, Ragul? What is gene? Yes, Ragul.
Ma'am, it is something similar to parents and.
In the last class, we had discussed about some terminologies, right? Did you note that?
No, ma'am.
Why? Why, Ragul? Yes, Ragul, tell me. I'm waiting. Jason, what is gene? Take out your notebook, Ragul, and start writing. Yes, Jason. What is gene?
It is information stored inside cell which controls synthesis of proteins and like synthesis of proteins. It helps in DNA for synthesis of proteins, and it determines the height or the inner characters of a offspring one.
Okay. Anthony, what is gene?
Ma'am, can you hear me, ma'am? Ma'am? Unit of inheritance segments of DNA that controls the expressions and the character.
So many definitions in one. Gene is the segment of DNA.
Controls the
That is,
expression.
yes, which controls the expression of any trait or expression of any character,
Yes, ma'am.
right?
Yes, ma'am.
So each and every character is basically controlled by what? Genes,
Gene.
right? And each gene is made up of pair of alleles, right? So inheritance of gene means particular gene who is carrying the information related to particular trait is transferring from one generation to another generation, and in the next generation the gene is actually expressing that particular trait. So there are two types of inheritance of gene. One is inheritance of one gene, okay, inheritance of one gene, and another one is inheritance of two gene, which is also called as I will tell you once we will start talking about the inheritance of two gene. So here one gene is made up of pair of alleles, right? As there is a transferring, or the single gene is actually controlling the trait, so that kind of inheritance is called as monogenic. It is called as monogenic inheritance. Monogenic. Mono means single. Okay. So in monohybrid cross, we always talk about one gene inheritance or inheritance of one gene, that is monogenic inheritance. Okay. So Mendel did the experiment, and as per their experiment, as per their observation and inference, he postulated the laws of inheritance. Okay. And what are they? The law of dominance and the law of segregation. Here we are talking about monogenic inheritance. Okay. Law of independent assortment is related to the digenic inheritance, where we are talking about inheritance of two genes. Are you getting me? So as we are talking about the monohybrid cross, monogenic inheritance, so we will include only these two laws. Okay, the law of dominance and the law of segregation. So let's see the law of dominance. Dominance means what? What is the meaning of dominance? Yes, what is the meaning of dominance? That person is very dominant. She is very dominant. He is very dominant, right? What is the dominant? Even in your friend circle, one of your friend is definitely dominant, right? He or she always feels that everybody should listen to her or he, right? So what? She or he is dominant. Okay. So law of dominance says that during this In the last session, we had discussed about the homozygous and heterozygous, right? So in the combination, in the hybrid gene structure, that is heterozygous condition, in the heterozygous condition, if one allele is expressing themselves completely and masking the other's expression, okay, that particular allele is called as dominant allele. Okay, so law of dominance says factor in a pair of allele, one allele is the dominant who is expressing themselves 100% while masking the other's expression. Okay, so in homozygous condition as well as in heterozygous condition, if any one of the factor is expressing themselves completely, that particular factor will be called as dominant factor. Okay, so in the cross you can see when we cross the first let's take an example of homozygous condition. Okay, suppose the pure breeding or the pure line is as a capital T for tall and small t for dwarf. So the F1 generation all will be the hybrid, right? Capital T and small t combination. And what will be the phenotype? Tall. Okay, which means the capital T homozygous condition, the phenotype was tall and here the phenotype was dwarf. After the combination or after the fertilization, we will get the hybrid structure, hybrid genotype, that is capital T, small t. And in the heterozygous condition, you will see all the plants showing their length as a tall, which means this capital T is expressing themselves completely while masking the expression of small t. So we can say that law of dominance is followed in both the situation, homozygous as well as in heterozygous condition. Clear? Law of dominance? Hello?
Ma'am, can you explain again? Ma'am?
Yes. Hello?
Ma'am, can you explain law of dominance again, ma'am?
Okay. Chalo, I am giving you one question. Okay. We have two plant, the parent plant, pure, round, okay, and wrinkled. You have to tell me the phenotype of F1 generation and F2 generation. Yes, everybody. Do the crossing. The round will be represented by capital R and the wrinkle will be represented by small r. Okay. What will be the F1 generation? Yes, Anmoldeep. What will be the F1 generation? All round. Okay, and what will be the genotype? Capital R and small r. Okay. Capital R and small r. That means the progeny of F1 generation, they have a heterozygosity. Means the zygote is having a two different allele for single gene, right? So in this situation also, what will be the phenotype? Round, right? That was for the homozygous condition also. And this represents that this round characteristic, the round trait, is actually the dominant, and this factor is the dominant factor. Why? Because this factor is expressing themselves completely, where the small r is not able to express themselves. Are you getting me, Anthony? Okay, now let's see the F2 generation. For the self-pollination in F1 generation. So let's cross. Let's make a gamete. So this capital R, the fusion will form homozygotes or the homozygote. Then this one will fuse with the small r and it will form heterozygosity, small r, capital R, it will form heterozygosity, small r and small r will fuse and form homozygosity. And you will see the phenotype, all three will show round, whereas this small r, which is in a homozygous condition, will show wrinkled seed. So this cross explains that this capital R is the dominant one, because in the presence of that, small r is not able to express, right? And this law of dominance explains that why all the progenies of F1 generation is having single phenotype. Why? Because of the presence of dominant factor. This law of dominance explains that in F2 generation, why there are 3 round shaped seed, whereas only one seed is wrinkled shape out of 4 offspring. Plus, this law of dominance also explains that in a mixed population, okay, in a mixed population, which one is superior? The dominant one. Why? Because this dominant factor is having the ability to suppress the other trait, and that suppressed one is called as recessive. Is it clear, everyone? Law of dominance and its significance? Okay. Now, the another law of inheritance is law of segregation. This law of segregation you will always read, and you have always seen in the book after the law of dominance, right? You always have to study about the law of dominance and then the law of segregation. But law of segregation is the first law of inheritance. Why? Okay. So, for the inheritance, for the recombination, first the parent have to produce gamete, right? And the gamete have the ability to transfer the gene from the parent to the offspring, right? Because offspring is made up of zygote, single cell, and zygote is made by the fusion of gametes, right? This law of segregation actually focused on the formation of gamete, and that's why it is the first law of inheritance. Clear? Though we always used to study after the law of dominance in the second position, but still it is a first law of inheritance. Is it clear, everyone? Okay. So the parent, whether it is a heterozygous or homozygous, they first have to produce gamete. For example, here the yellow color seed. Okay, so it is a heterozygous parent. Male is heterozygous. Capital Y is representing here the yellow color, and small y is representing here as a green color. So let's cross with the female parent, which is again the heterozygous. Okay. So the male parent and female parent first have to produce gamete, and during the gamete formation, these alleles, the pair of alleles have to split in such a way that all the gametes will get equal amount of allele for a particular gene, for a particular trait. Okay, so this law of segregation says that during the formation of gamete, the pair of alleles splits and segregate without affecting each other. Okay, are you getting me? So during the formation of gamete, how the gamete will produce? By the process of meiosis, cell division. So the genes will split or segregate at which point of meiosis? Anaphase 1. Okay, so at anaphase 1, during the gamete formation, the alleles of particular gene will segregate without affecting other allele of the same gene. Are you getting me? And then by completing the whole process of meiosis, the gamete will get particular allele from the particular gene. Each gamete will carry each allele for the particular trait. So this the parent with the capital Y and small y will produce the gamete, and each gamete will have, one will have small y, other one will have capital Y. The same with the female also. One gamete will have capital Y and another gamete will have small y. So this segregation of allele during the gamete formation is called law of segregation. This law of segregation is also called as law of purity of gamete or spore. Why it is called as purity? Because each gamete is having a single allele for each and every trait. Are you getting me? As there is no mixing, no recombination, no hybrid, and that's why it is called as law of purity. As there is no hybrid in the single gamete, you will find single allele for particular gene. Okay, and that's why it is called as non-mixing of allele. If the gamete will have allele like capital Y and small y, then in this condition we cannot say that it is a law of purity of gamete, because the gamete is having both the type of allele for single trait. Plus it will not be called as non-mixing, because there is a mixing of two different allele for single trait. Are you getting me? Why the law of segregation is called as law of purity of gamete and law of non-mixing of allele? Yes, everyone? If yes, then please do like this. Okay, what about you, Jason? Okay, Ragul? Okay. Now the next law is law of independent assortment, that we will study while we will talk about the dihybrid cross. Okay, so here let's take an example of tall and dwarf plant. As you can see, the parent plant is producing gamete, and each gamete is having a single allele for particular trait, right? And in the F1 generation, all the offspring will be hybrid with single type of phenotype. While we do the selfing of the F1 generation, then we will get the different types of phenotype. The tall with the homozygous condition, tall with the heterozygous condition, and dwarf. So what will be the phenotypic ratio and genotypic ratio? Phenotypic ratio will be 3 tall, so 3:1 dwarf. So 3:1 is the phenotypic ratio for monohybrid cross for law of dominance or segregation. What will be the genotype? Only one type of progeny is having capital T combination. So let's write it as 1. 2 progeny is there, like they have a hybrid genotype, capital T, small t, and so it is 2. Then only 1 progeny is having homozygous small t combination, so it is 1. So what will be the genotypic ratio? 1:2:1. Dihybrid cross means cross between 2 characteristics, 2 traits. While crossing, if we are considering 2 traits at a time, then such kind of cross is called as dihybrid. 2 traits. For example, if we are considering the shape of seed and color of the seed, okay, during the crossing, as we are considering 2 traits, so it will be called as dihybrid cross. If we will consider 3 traits, then it will be called as trihybrid cross. Okay? Dihybrid cross, clear the definition? So, we have a parent like yellow seed, and another parent is wrinkled and green seed. When the Mendel was doing their experiment, he has selected the 7 contrasting pair of traits, right? In that, 1 trait is dominant, whereas the other contrasting one was the recessive, right? So for the color, yellow was the dominant, green was the recessive, shape, round was the dominant, wrinkled was the recessive, height, tall was the dominant, dwarf was the recessive, right? Pod shape, pod color, yes, the yellow, green, inflated, right? Okay. So let's see the dihybrid cross. As you can see here, we have a parent round yellow seed, and another parent is wrinkled green seed. So first they have to produce gametes. So how they will produce? They will produce 2 different types of gametes. One combination will be capital R, Y, capital R, capital Y. Another combination will, another parent will produce small r, small y gamete, small r, small y gamete. Right? Suppose we have a parent like capital R, small r, capital Y, and capital Y. Then how many gametes will produce from this parent? How many gametes will produce? For this, there is a very short trick. 2 is to the power n. Okay?
Ma'am, it's capital Y and capital Y, ma'am.
Sorry?
Is it capital Y and capital Y or capital Y and small y?
For what you are saying?
Small
This one?
Yes, ma'am.
Capital R, small r, capital Y, small y.
Okay.
First, we need to know how many gametes will produce in what condition. Homozygosity, heterozygosity. Okay. So, for that, we have a formula 2 raised to the power n. Because in exam, in NEET exam, so many times examiner will ask you, they will give you the combination, the genotype of the parent, and they will ask you the number of different types of gamete. Okay, so how you will get to know? By making the different combination, it will take huge time. So for that, go with the formula 2 raised to the power n, where n is what? Number of heterozygosity. Number of heterozygosity. Okay. This is homozygous or heterozygous? this is homozygous or heterozygous? Please tell me fast. Oh, capital Y is a heterozygous. Homozygous. This is heterozygous or homozygous? Hetero. That means,
Ma'am,
what is the value of N?
ma'am,
Yes.
is it capital Y or small y or capital Y or capital Y?
This is capital Y or what? This is what? Capital Y? Because I used to write small y like this.
Okay, ma'am.
Are you getting me?
I think it is capital Y and small y, so that's
No.
hetero.
Yes. So what will be the value of N? 1. So 2 raised to the power 1 means this type of parent will produce 2 types of gamete. Right? Which means capital R, capital Y wala combination and small r and capital Y wala combination. Is it clear? Chalo, I am giving you one more question related to this. We have a parent like A, capital B, small b, capital C, capital C. Now you have to tell me the number of types of gamete. How many types of gamete will produce by this type of parent? Very good. 2. Why? Because only one is the heterozygous, right? And other 2 are homozygous. Chalo, I am giving you one more. Now tell me. Types of gamete. Types of gamete and not the number of gamete, okay? Different types of gamete. 4. Yes, because the number of heterozygosity, that is N, is equal to 2. So it will be 4. So this concept is clear to you, how you have to calculate the types of gamete just by the genotype of parent, whether it is a heterozygous, homozygous. Is it clear, everyone? Okay. So this is what, this is actually law of segregation for monogenic, law of independent assortment for, yes, digenic. Yes.
Ma'am, can you write the gametes which will be formed in the second case, ma'am?
Okay. So let's take example of this. So they will produce capital A, capital B and capital C type. Another gamete will be capital A, small b and capital C type. For this, one gamete will be capital A, capital B and capital C type. Another gamete will be small a, capital B and capital C. Another gamete will be capital A, capital B, small c. Another gamete will be capital A, capital B and small c. Is it clear, everyone? Is it clear?
Yes, ma'am, it's clear.
So basically you have to make a combination. Yes, Jason. Are you getting me? Hello, Jason. Okay. Bhuvana. Anthony, Shashidharan, Prashanti, Kirtini, Kevin, everybody, are you getting me? Chalo. So we were talking about dihybrid cross. So the parent is capital R, which is a dominant, round and yellow. And another parent, the pure line, small r, small y, which is wrinkled and green shape. So they will produce the gametes like capital R, first, if it is the pure line, then it will make capital R, capital Y, capital R, capital Y wala combination, and same with the small r, small y, small r, small y. If the parent is heterozygous, then the combination will be capital R, capital Y, capital R, small r, capital Y, small r, small y. Okay, the same with this parent also, the round and yellow, which means we are doing the self-pollination, selfing, or the self-fertilization. And by the crossing or selfing between them, they will produce F2 generation. In the F2 generation, we will get different combination in the hybrid and in the heterozygous and in homozygous condition. So we will find 9 progenies out of 16, which is round and yellow, completely dominant, wrinkled and yellow, hybrid, but here the dominant factor yellow is there. Then round and green. The total number of round and green progeny is about 3, and only one progeny is there which is completely recessive, or we can say that heterozygous recessive trait. Are you getting me? By the dihybrid cross, we will get total 16 progeny, okay, out of which the phenotypic ratio will be 9:3:3:1. This is what? Phenotypic ratio. Genotypic ratio? What will be the genotypic ratio? What will be the genotypic ratio of dihybrid? How we will calculate that? The genotype. Yes. What will be the genotypic ratio? Who will say? Yes, Jason will answer. Come on, Jason, tell me the genotypic ratio of dihybrid cross.
Cross wheel for in monohybrid cross wheel. Ma'am, are you there?
Sorry. Yes, tell me.
We did the genotypic ratio as
yes, tell me. R, capital R, Y, Y, one combination. So how many progenies are there with this combination? 1. Okay, then we have a capital R, capital Y, and small y combination. 1, 2, only 2. Then we have a capital R, small r, capital Y, 1, then 2, then 2. Then we have a combination like capital R, small r, capital Y, small y. How many of this combination are? 1, 2, 3, 4. Then we have a capital R, small y, small y. 1, only 1. Then we have another combination, heterozygous and round and pure green. So 1 and 2. Then we have a homozygous wrinkled and heterozygous yellow. That is 1. Then we have a homozygous wrinkled and dominant yellow 1. And pure recessive one is 1. So what is the genotypic ratio? 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 1 is to 1. So mostly for the dihybrid cross,
Ma'am, for
examiner
that
will
smaller capital Y, small y, it's 2, ma'am.
sorry,
Ma'am,
small
for
r,
smaller, capital
and
Y and small y.
small. I didn't mention that.
Ma'am, you wrote us 1, ma'am.
Where?
Ma'am, at the bottom of the slide, like you wrote for smaller, capital Y, small y, you wrote us 1, but there is 2, ma'am.
Sorry, 2. Yeah. So this is the genotypic ratio. In the exam, for your board exam, the examiner will ask about the genotypic ratio of dihybrid cross. But in NEET exam, they will always ask you about the phenotypic ratio because genotypic ratio is huge, right? So mostly they will not ask about the genotypic ratio, but you have to know. Okay? So we have total 16 number of progeny from the dihybrid cross, and these are the number of plant, phenotype and genotype that we have already discussed. Now the next is law of independent assortment. Okay. What is law of segregation, Anthony? Law of segregation. Yes. Yes, Anthony. What is law of segregation?
Ma'am, two alleles of a gene controls each other. Ma'am.
Sorry?
Two alleles of a gene controls each other.
Alleles of a gene controls each other. Is it so?
Each character.
Each character. Okay.
To stay together, ma'am.
Okay. Okay, in front of you, there are the genotype of parent. Now you have to tell me the gamete. Tell me the gamete of genotype of the gametes produced by these two parents. Yes, Anthony. Gametes produced by this parent, gametes produced by this parent. Tell me.
Capital R and capital Y, ma'am. Capital R and capital Y.
Capital R and capital Y.
Capital R and capital Y.
Okay, what about this?
Small r, small y, small r, small y.
Why there is small r, small y? Why not capital R, capital R? Why not capital Y only? One gamete should have only one type of gene, na?
Yes, ma'am.
Then, but the gamete is having allele from both the genes. Yes, Anthony. Did you study cell division in class 11th?
Yes, ma'am.
Can you tell me the types of cell division?
Meiosis and
Meiosis and
mitosis.
mitosis. What is mitosis?
Mitosis. Ma'am, mitosis is multiplication.
Sorry.
Multiplying, ma'am, mitosis.
Okay. And meiosis?
Dividing
Dividing of what?
two cells.
Then why mitosis is also called as cell division? If meiosis is a cell division, if in meiosis cell is dividing, then why mitosis is called as a type of cell division method?
Ma'am, I don't know.
Huh? How can you say this? Kirtini. What is meiosis, Kirtini? Hello, Kirtini.
Ma'am,
Yes, Kirtini.
because it is dihybrid.
Huh? I asked you what is meiosis.
Oops. Ma'am, it is an reduction division, ma'am. So it divides into two, ma'am.
Okay, and what is mitosis?
Equational division, ma'am. If it has a 23 chromosome, the offspring also carries 23 chromosomes.
And can you tell me meiosis with respect to number of chromosome only?
Ma'am, meiosis,
If a parent is having 46 number of chromosome, after the meiosis, offspring will have
23, ma'am.
that means the number of chromosome is getting half, right? And
Yes,
that is
ma'am.
not offspring, that is gamete. Okay. In animal cells, meiosis type of division takes place only and only for the production of germ cells, that is gamete. Okay. And rest other cells are divides through mitosis only, right? So as we know that meiosis is a reductional division, that means here the number of chromosome is getting reduced, right? And gene is present in the, what is the location of gene? DNA. And what is the location of DNA? DNA get coiled, highly packed and form a prominent structure, that is chromosome. So if the parent is having chromosome, okay, suppose the parent is having 2 chromosomes. So during the meiosis cell division completes into 2 stages, meiosis 1 and meiosis 2. So in meiosis 1, they will make 2 daughter cell, right? And by the meiosis 2, they will make 4 daughter cell, and each will carry each chromosome, right? Right? So gene, it is not necessary the pair of alleles for one gene is present only one chromosome. It can be present in homologous chromosome. Are you getting me? So during the meiosis cell division, anaphase 1, the alleles will segregate from the pair and they will come and packed into the gamete. So one R will come and pack into one gamete, and one capital Y will come and pack into the same gamete. Like this, each gamete will have half of the DNA or half of the gene from one parent, so that after the fertilization, both the half will fuse and make a total 46, which is a normal number of chromosome of human, right? Right? So the same thing is going on here also during the formation of gamete. And this is law of segregation. While sorting, while segregating, while separating, okay, this particular allele will not affect the separation of this particular alleles. And this is what? This is law of independent assortment. The alleles of each trait will segregate, will separate independently without affecting others or without getting effect from other. This is what? This is law of independent assortment. Law of independent assortment and law of segregation is somewhat similar. But the thing is, we cannot say that this is a non-mixing allelic law of non-mixing. No, we will use only for the segregation only and not for the law of independent assortment. Till now it is clear to everyone? Pakka? If yes, then do like this, everybody. Okay. What about you, Anthony, Ragul? Yes, Anthony, is it clear to you? Law of segregation and law of independent assortment? Pakka? Now tell me, what is meiosis? Anthony. Oh God, again meiosis. Yes, Anthony. Just now we did whole story. Yes, Anthony, make it fast. Anthony, write down. What is meiosis? What is the difference between meiosis and mitosis? You are going to teach us these two topics in next class. Okay, Anthony? You already studied all these things in your class 11. So you have to know. Okay? So law of independent assortment is what? Is the segregation of alleles of a particular trait does not affect the segregation of other alleles, segregation of alleles of other trait. Or we can say that they segregate independently in the case of inheritance of two genes. So, please go through the statement of law of independent assortment. This is called as second law. Actually, Mendel did the experiment to prove that something is passing from one generation to next. Then why only this character is getting expressed throughout the offspring in one generation, and why suddenly in next generation some of the characteristic is expressed by getting expressed in n number of organisms. Why in one organism parent type character that is recessive. That was postulated or given by the Mendel as a law of dominance and segregation. But this was not enough. So few of the scientists, not few, the three scientists came up and they said they rediscover the Mendelism or the Mendel's experiment, and they said that there are still something that happens or that is happening throughout the process. And then they said law of independent assortment, because Mendel did the monohybrid cross. He never did the dihybrid cross. Okay, so these three, the Hugo de Veris, Carl Correns, and Eric von Teschmark, these three scientists, inspired by the work of Mendel, and he rediscovered, and then they said that yes, Mendel is the father of genetics, but there are still something missing, which is the Dihybrid cross. So they did the Dihybrid cross, and they said the law of independent assortment is the second law of inheritance. And who said the particular statement? Karl Correns. Clear? Karl Correns said that law of second law, because law of segregation was also saying the segregation of alleles in monohybrid cross. Law of independent assortment says segregation of alleles in digenic inheritance, in two gene inheritance. The same thing is actually explaining in both the law, but the condition is different. One is monohybrid and another one is dihybrid, and that's why it should be the Mendel's second law. Okay. So the law of independent assortment is clear, everyone? Now look at here the cross, the example of that. Here we have a pure line parent. Okay, I think we did this cross, so there is no need to explain it again. Now I am going to give you a problem on this topic. We have a parent like white and purple color flower with white and purple color flower with the round shaped seed. Now you have to tell me the type of gamete. First you have to tell me types of gamete they will produce. Let's see.
Ma'am, is it for pea plant?
Sorry?
Is this for pea plant, ma'am?
Yes.
But isn't the violet color the dominant in pea plant? So why is W used here?
Here the white and purple.
Yes, ma'am. The violet is the one which is dominant. So shouldn't be the letter be V?
We can use anything. Let's take a V also. So here let's take violet, right? V. Actually, whenever we select the color of flower, right? The white and purple, right? And you are saying white is the recessive one, right? So when we do the crossing, creates confusion. So it's better to write W. But for your convenience, let's write V. Okay? V and round and V and wrinkled. Now it is perfect. Now you have to tell me the types of gamete. How many types of gamete will be produced by this parent 1 and parent 2? Yes. Bhuvana, please keep your camera on throughout the session, okay? Yes. How many, what are the different types of gamete produced by P1 and P2? Only one type. Perfect. Now we have to tell me the phenotypic ratio of crossing between these two parents. Yes. What is the phenotypic ratio? What is the phenotypic ratio? Whatever the combination will be, whatever the types of gamete will be, the phenotypic ratio of the dihybrid will always 9:3:3:1. Okay. Okay, everyone. So this is all about the law of inheritance. That is law of dominance, which is not the first law. Law of segregation, which is the first law. Law of independent assortment, which is the second law. And dihybrid cross. Okay, now let's take a small break and then we will discuss about the next topic, gene interaction. Okay, so let's meet after 5 minutes. Hello. Hello. Whatever we are discussing, and are you getting everything? All three laws, dihybrid cross.
Yes, ma'am. Actually, this was taught in 10th, so it's not that difficult to get it.
Okay, good. Others are still enjoying their break. Okay, Bhuvana, I understand. Okay, next topic is gene interaction. Till now, we were discussing about inheritance of gene, right? Particular gene for particular trait is getting inherited from parent to offspring, right? Now, the next is how the alleles of the same gene or different gene, they interact with each other and give the expression. And this is what gene interaction. What is interaction? Here we are discussing something. We are interacting with each other. So same way, the allele for same trait or the single gene or for the different gene, right? If they interact with each other in terms of expression, okay, then this is called as gene interaction. So there are two types of gene interaction. One is intergenic and another one is intergenic. Intergenic means if the alleles are interacting with each other. For example, here the alleles, both the alleles are interacting with each other and giving the response accordingly. So it will be called as intergenic. Intergenic, just like inter-college, inter-school competition between the two school, right? So the same thing, if the interaction is taking place between the two genes, okay, and then it will be called as intergenic. And that's why we never use the term allelic interaction for intergenic, because here the two genes are actually interacting with each other, and not the single gene, not the single allele is doing that. Okay. So the best example for intergenic is incomplete dominance. Here we can see the red flower, which is a dominant color, and white flower, which is a recessive color. Okay. So while crossing these two, the alleles, by mixing, okay, and making the combination as capital R and small r. So here in the hybrid or the heterozygous condition, both the allele will interact with each other, okay, and they will produce some secondary or the intermediate characteristic, intermediate color. Okay, here not a single allele is there who is actually completely dominant or who is completely getting masked by the other. No, both are giving their response 50-50%. So from the 50 from this, from the 50 from this, they mix together and make us something new, which is called intermediate, in between these two. Okay, so this type of interaction between the allele in same gene is called interallelic or intragenic, inside the same gene between the two allele. Are you getting me? So here, by this interaction, both the gene will give their 50%. So by giving the 50% from both the character, they will make some intermediate character, which is pink color, combination of red and white. Okay. And here not a single allele, no one is actually completely dominant. So it is called as incomplete dominant. Both the trait, both the character, both the color is actually incomplete dominant because they both are giving their response. They both are getting expressed partially. So it is called as incomplete dominance.
So in this case,
sorry,
the laws of inheritance do not actually be followed, ma'am, in these cases?
Yes, the Mendel's law of inheritance, Mendel's actually never said that there is something who will express partially or who will suppress others partially. No. All those inheritance who does not follow the Mendel's law, dominance, segregation, and law of independent assortment, they all are called as neo-Mendelism. Means after the Mendel. Okay, all those new type of inheritance which is not considered by the Mendel, they are called as neo-Mendelism. Whether it is a incomplete, whether it is a codominance, whether it is a multiple allelism, pleiotropy, epistasis, polygenic, every type of inheritance apart from the Mendel's one, they are called as neo-Mendelism or non-Mendelism. Okay, so Mendel never said that there are some characteristic of gene where they can interact with each other or getting influenced from other. He never said this. He always said something is there who is passing from one generation to another and getting expressed. That's it. So please make a note. What is neo-Mendelism? Because these terms you will not find in your NCERT. But you will get question like neo-Mendelism, non-Mendelism.
Ma'am?
So you have to know what is neo-Mendelism, what is non-Mendelism.
Ma'am? Ma'am, can you please spell it, ma'am?
Neo.
Okay, ma'am. Got it.
Ah.
I heard it as something else, ma'am.
Or non-Mendelism. So let's take first example of neo-Mendelism or the example of intragenic or interallelic interaction is incomplete dominance. Here in this type of inheritance, both the allele have the ability to express 50%, and that's why new characteristic will get formed in the offspring, which is the intermediate in between both the parents. Okay, the main and very important characteristic of incomplete dominance type of inheritance, their genotypic and phenotypic ratio. Okay, this is the only one inheritance where the phenotypic ratio and genotypic ratio both are equal. Let's take an example of snapdragon. Here the flower is red, the dominant flower is red and recessive flower is white. So while crossing these two, the F1 generation, all the plants will have, they will show incomplete dominancy, which is they will produce intermediate characteristic, that is pink. Okay, this is about F1 generation. But in F2 generation, while we do the selfing of this pink color, they will produce different types of offsprings as well. So some are red in color, two offsprings will be pink in color, and one will be white in color. So what will be the phenotypic ratio? 1:2:1. And what about the genotypic ratio? Homozygous red color RR1, heterozygous pink color which is 2, and homozygous or the recessive one is 1. So you can see that the phenotypic ratio and genotypic ratio both are equal. Okay, this is very important. Please make a star or note, because there is a 99% chance that you will get question from this concept. Another example of incomplete dominance is 4 o'clock plant. The same red, if we will cross red and pure white, we will get hybrid pink color. And if we will do selfing of this pink color flower, we will get same phenotypic and genotypic ratio. Now let's see in animal. This is Andalusian fowl, where one parent is completely black and another parent, the chicken, is white. While crossing these two, we will get blue color chicken. Okay, this blue color chicken will have the feather with stripes. The stripes will be like white and blue in color. Sorry, white and black in color. Are you getting me? At least say yes or no. Only N and Sarjana. What about others? So when we cross these two pure form of chicken, the black and white, we will get blue color due to what? Due to incomplete dominancy. But this blue color bird will have alternate black and white stripes on the feather. Okay, and when we do selfing in this blue colored fowl, we will get black, two blue color, the same of with the parent, and one is recessive. So same the phenotypic ratio and genotypic ratio will be 1:2:1. This is the example for animal cell. So here we had discussed about 3 examples of incomplete dominance, which is in snapdragon, which is in 4 o'clock plant, and Andalusian fowl. Now let's see some questions related to this. In case of incomplete dominance, monohybrid F2 dash is 1:2. What about others? Kirtini said A. Okay, Kirtini, please explain me how the answer will be option A. Yes, Kirtini. Kirtini, please explain me. Hello, Kirtini.
It is monohybrid, ma'am. So in F2 generation, it has genotype.
Okay, in monohybrid cross. And what about incomplete dominance? Monohybrid means, Kirtini? Monohybrid means?
One gene is going to pair with another.
Okay, so if we are considering one character, that is height, is it a monohybrid cross? Kirtini, am I audible?
Yes, ma'am.
Is it monohybrid or dihybrid?
Monohybrid, ma'am.
Okay, if I'm selecting it as a capital T plus round and small t and wrinkle, Is it dihybrid or monohybrid?
Dihybrid, ma'am.
Okay. Then we had selected the color of flower. Red color flower crossed with white color flower. Is it monohybrid or dihybrid?
Monohybrid.
Monohybrid. Okay. So after crossing or doing this monohybrid crossing, we are getting this type of genotype, right?
Yes, ma'am.
So as per your knowledge, in monohybrid cross, what will be the phenotype of this offspring?
Phenotype, ma'am.
Yes, phenotype.
Uh, 1:2, 2:1.
Phenotype. I am not asking about the ratio. Just tell me the phenotype of this particular hybrid plant.
Tall, pure round, ma'am, and
Hmm. Okay. What will be the phenotype of this hybrid? Yes, Kirtini. Answer it quickly.
capital T, capital T.
Phenotype. Please tell me phenotype. Ragul, what is the meaning of phenotype?
Ma'am,
Yes, Ragul. I was keep on asking, are you getting me? Is it clear? At that time, you people said, yes, it is clear. I got everything. And once I start asking the question, you people are like, hmm. Throughout the session, we were doing crossing, right? And throughout the session, we were identifying the genotype, genotypic ratio, phenotypic ratio, right? At that time, why you people didn't ask me, what is this? Why are you asking? I don't know what you are saying. Ragul and Kirtini. Bhuvana. What is phenotype, Bhuvana? What is phenotype? Madhumita, what is phenotype, Madhumita?
Ma'am, the appearance like color, shape, like that, ma'am.
Anthony, what is phenotype? Don't search here and there. No, I will not accept if you will answer from your textbook or notebook.
ma'am, I will. 1 minute, give me, ma'am. I will think, ma'am. Ma'am, what is the question actually, ma'am? Phenotype.
Oh, then what are you searching?
No, I lost my connection, ma'am. Just now I came back, ma'am.
Oh, is it?
Ma'am, seriously,
Okay.
ma'am.
I got it.
Ma'am,
Okay. What
what's
is the
the-
meaning of phenotype?
Ma'am, the physical appearance of
Yes. Phenotype. Physical. Physical appearance of any organism is the phenotype. Okay. Now Kirtini will tell me the phenotype of this particular plant and this particular plant. Yes, Kirtini, tell me.
Ma'am, 3 tall and 1 short,
Just
ma'am.
tell me phenotype of this plant. No problem, Anthony.
Tall, ma'am.
Tall. Very good. Now tell me the phenotype of this particular plant.
Round, ma'am.
Round.
Yes,
But
ma'am.
I said that the phenotype of this plant is red and this plant is white.
Red, ma'am.
Red, sure.
Yes, ma'am.
Okay. But as per neo-Mendelism, what should be the phenotype of this plant? Yes, Kirtini. Ragul.
Get it, ma'am?
Yes, Ragul. What should be the phenotype of combination of capital R and small r?
I'm 3:1.
Huh? I am asking phenotype and you are saying the ratio.
I'm red, ma'am.
Red.
Pink, ma'am.
Pink. Yes, pink. Why? Because they are incomplete dominant. So question is what? Incomplete dominance. Monohybrid, Kirthini. This cross is again monohybrid because here we are considering only one trait. So it is monohybrid as both the allele have the ability to express themselves, and that's why it is incomplete dominance. And the characteristic of incomplete dominance heritance is the phenotypic ratio and genotypic ratio both are equal, and that's why the answer will be option C and not A. Now it is clear to you, Kirthini?
Yes, ma'am.
Pakka?
Yes,
What
ma'am.
about you, Ragul? Is it clear to you? Sure? Okay. Next question. A pea plant with yellow and round seeds is crossed with pea plant having green and wrinkled seeds. Then in F2 generation of this dihybrid cross, 320 plants are produced. Means total number of plant is what? 320. Out of which 180 plants have same phenotypic characteristic. Identify this phenotype. So first you have to make a crossing between these two. The total ratio is like 9:3:3:1, which means total 16. So here the question is what? This ratio should be equal to 320, right? This is what? Phenotypic ratio. So now you have to tell me the type of phenotype of 180 plants. I already gave you the hint. if you have seen in the dihybrid cross, in the Punnett square, there was about 16 blocks, right? Which means they are representing 16 offsprings. But here the number of number of offspring is more, which is 320. Which means in each block, how many offspring will be there? You have to divide, right? 320 by 16. That is 20. So from these ratio, you have to see which characteristic will be present in 180. Kirtini said yellow and round unmolded, and 3 of them is saying option B. What about others? Sarjana, Bhuvana, Kirtini, Prashanti, why are you not answering? Ragul. Okay. What about others? Sindhuja, Shashidharan, Gopinath. The answer will be yellow and round. Okay. You do not have to do anything. Just you have to see 320, 180, and you already know the phenotypic ratio 9:3:3:1. So by that you can find the phenotype of 180 offspring. Okay, so let's see. The first law of Mendel is law of inheritance, law of variation, law of independent assortment, law of segregation. Yes, Ragul. What will be the answer, Ragul? Yes, Ragul. Tell me the answer.
D, ma'am.
Sorry?
Option D, ma'am.
Why?
Yeah.
Why not option A, law of inheritance? Tell me. What about others? Yes, what will be the answer? Option D, law of segregation. But Ragul, the question is same. Why not law of inheritance? Sarjana. Yes, Sarjana.
Ma'am, because the law of inheritance, there are 3 laws, ma'am. Law of dominance, law of segregation, law of independent assortment. Here it is asked first law of Mendel, which is law of dominance, law of segregation. Law of dominance is not there in the option, so I went with option D, law of segregation, ma'am.
Chalo, I am giving you the option. Law of dominance.
Ma'am, but the first law was law of segregation, ma'am.
But you said law of dominance and law of segregation. And now you are saying law
No,
of...
ma'am, second law, like first law is law of segregation, ma'am.
Okay, and what is the second law?
Law of, second law of, like, ma'am, are you asking about the law of independent assortment, which is not given by Mendel, ma'am? It
But
was
still
second law of Mendel.
It is second law. Law of independent assortment is the second law of inheritance, which is said by the Corins. The first law is law of segregation, which is again said by the Corins. Mendel never said that this is the first law, this is the second law, this is the third law. Mendel never said. First, second, third position was given by these 3 scientists. Okay?
Yes, ma'am.
They are the only one who said that This is the this should be the Mendel's first law. This should be the Mendel's second law. Clear?
Yes, ma'am.
Okay. So the correct answer is option D. Next question. In dihybrid cross involving two genes A and B, F1 heterozygote is crossed with homozygous recessive parental type, which is capital A, small a, and small b. What would be the ratio of offspring in the next generation? So it should not be the A and B. It should be capital A, small b, like this. From this question, you can identify that they are talking about test cross, right? Right? So here it is heterozygote is crossed. Heterozygote means capital A. It may be the capital A, small a, and capital B, small b. So heterozygote will crossed with recessive parent, which is small a, and small b, So now you have to tell me. Yes. Are you getting the question? What they are asking? Indirectly, they are saying that this is the test cross, and in test cross, we with the help of test cross, we used to identify the genotype. Genotype, right? That means in this question, you have to identify the genotypic ratio. What, Sarjana? Examiner will not ask you directly, okay? They will hold like this. Now you have to cross and fill, cross and fill, cross and fill, cross and fill. And tell me the ratio? Oh, as per Bhuvana, the answer is option D. Chalo, let's see. Yes, Ragul, what will be the answer? First, Ragul will tell me the genotype of offspring. Okay? Okay, Ragul? Chalo, do the crossing and then tell me the genotype of offsprings. Is it done, Ragul? Say yes or no. Done? Chalo, tell me here. Yes, Ragul, tell me the genotype of offspring. Small b, capital A, capital B. What will be the genotype? The combination? Am I audible, everyone? Am I audible? Am I visible to everyone? That means Ragul can also hear me and see me, right? Ragul, tell me. Yes, what will be the combination here? Tell me.
Capital A, capital B, small b.
Okay, what about here?
Small a, capital B, small b.
Okay, what about this?
Small a, small b, small b.
Okay, what about this?
Capital A, small b, small b.
Now tell me the genotypic ratio. Yes, Ragul, genotypic ratio, tell me. We don't have much time. Do we have same combination? Or we have only one combination of this type? Capital A, capital B, small b. Do we? No, only one type, right? What about this, Ragul?
One, ma'am.
One. What about this combination?
One.
What about this combination?
One, ma'am.
Okay, this is what? This is genotype. Here we are talking about genetic makeup. Genetic makeup means the capital A, capital B, small b, they are representing allele and their combination is representing here as a gene. So they are the genetic makeup and the ratio is 1 is to 1 is to 1 is to 1. Clear? So Ragul, tell me which one is the correct option?
C, ma'am.
Pakka?
Yes, ma'am.
Okay. So the correct answer is option C. So in today's class, we had discussed about inheritance of gene, that is law of inheritance, law of dominance, law of segregation, law of independent assortment, right? After that, we had discussed about the gene interaction. What is gene interaction, Bhuvana? Yes, Bhuvana, what is gene interaction? Sarjana, what is gene interaction?
Ma'am, it is the interaction between the same gene, but it is the interaction between different alleles, ma'am.
Okay, there are two types of interaction, right?
Yes, ma'am.
What are they?
Ma'am, it is intragenic, ma'am, which is like not and non-allelic, non-allelic.
Intragenic and intergenic.
Yes,
Ah.
ma'am.
Okay, and Moldeep, what is intragenic interaction? Intragenic interaction. Intragenic interaction means within the same gene, which means there is an interaction between the alleles of the same gene. And that's why it is called as intra-allelic interaction. Okay. Another type is intergenic, which means interaction between the two different genes, where the genes are actually getting involved and not the single alleles. This kind of interaction is called as non-allelic interaction. Okay. Yes, Anmolti.
Can you explain it again, please? Actually, I also don't.
Sorry?
Can you explain it again?
Interaction of gene means interaction between the genes so that they can give the expression. Okay, because sometimes what happens, one of the gene get influenced from the other gene, one of the allele get influenced from the other allele, and in such a way that some alleles will not be able to express themselves, whereas some will express completely, whereas they give the mixed expression. Okay, so there are two types of interaction. Intergenic means between the two gene, interaction between the two different gene. Intragenic, within the gene, interaction between the alleles within the gene. Are you getting me? So this type of interaction, intragenic, is called as interallelic interaction, because two alleles within the gene is getting doing the interaction, and one of the allele will get influenced, or likewise they will do the interaction and give the expression. Okay, and incomplete dominance is the example of this type of interaction. Clear? Okay, so this is all about today's session.